Some of our earliest triple point of water cells went to NIST to be evaluated, and the gentleman at NIST wrote a paper and published it saying that our triple point of water cells were of the highest quality worldwide. They were as good as anything.
It has been a few years since I've built them. I built things for Pfizer and Moderna, so that's kind of nice to know I was a part of like that whole process. And the process essentially starts with getting glass in from a manufacturer, and then we take that glass and we clean it. Cleaning process is pretty stringent. It involves acid cleaning and using lab-grade detergent, and it's a lot of soaking. And then after you do that kind of stuff, you move into steaming, and you steam the cell for about a week. And when we have an order, we take the cell and we distill water into it. The type of water that we use is called Vismo water, and it comes from a very specific section in the ocean. It's about 6 stages, I believe, of distillation, and we're getting all the salinity out of the water, all the salts being taken out. And then once the cell is filled, you hook it up to a vacuum system. And that part of it's kind of cool. People like to watch that. You use a torch and you open the vacuum system with the torch, and you connect the cell to the vacuum system with the torch. So the glass is actually attached to a glass vacuum system. And then there's a series of different pumps and gauges and knobs, and it's a lot of, turning and making sure that the pressure's just right. And then once you have everything right, you take it off with a torch, and which is sealing it. That's how you seal the pressure inside there in the vacuum. And then once you have that, you check it for its proper pressure, and you can do like an audio check. And if you do it just right, you can get like a little water hammer. I mean, that's in a nutshell how you build those.
A fixed point cell, one of a few different types of fixed point cells. That is what I use to calibrate my standards. I also use it to do interim checks. So in between batches of probes that I calibrate, I will use that triple point cell on my standards to keep track of drifts, deviations, and make sure that my standards are always within tolerance.